Hello my dear students. So in the previous chapter we discussed what classification is, and thereafter we came to the 3-domain classification. Under that we discussed the first 2 domains, Domain Archaea and Domain Bacteria. So I'm going to move on to the 3rd domain, Domain Eukarya. Now from the name Eukarya, there are organisms that are made up of eukaryotic cells. That's why the name Eukarya. Now you all can remember prokaryotic cells. What are they? Cells that do not have a well-defined nucleus. So prokaryotic cells are those. Then what are eukaryotic cells or eukaryotic cellular arrangement? They have a cell with a well-organized, well-defined nucleus. So you're familiar with the structure of cells. You all have discussed plant cells and animal cells. So you're familiar with those. And there you can remember the cells have a prominent, well-defined, organized nucleus. So those organisms are all known as eukaryotic organisms. So under domain Eukarya, the most prominent organisms with a eukaryotic cellular organization belong to this domain. So here we can say they have well-defined nucleus. That is what you need to remember. So they can live in different environments. So under this domain, we will be looking at the plants, animals, then the fungi and protista. So all these organisms, they have varying habitats. You can find them in the terrestrial environment, you can find them in aquatic environment, maybe the sea water, fresh water, and even in the atmosphere. On top of the trees as well. Birds, they live on trees, they have the aerial environment. So like that, you can find them in any environment, or we can say they can live in different environments. Thereafter, if we look at the property, they are not sensitive to antibiotics. So here you can remember they are not sensitive to antibiotics. If you can remember, we discussed domain Archaea. Those organisms are also not sensitive to antibiotics. But domain bacteria, they are sensitive to antibiotics. Again, domain Eukarya, the organisms are not sensitive to antibiotics. Even we come under domain Eukarya. We are not sensitive to antibiotics. Plants are not sensitive to antibiotics. Even animals, they are not sensitive to antibiotics. That's why we can take antibiotics as a method of cure against pathogens. So you all have to remember that, students. They are not sensitive to antibiotics. So under domain Eukarya, there are 4 groups. The first one is Kingdom Protista. Then we have Kingdom Fungi, Kingdom Plantae and Kingdom Animalia. The 4 different kingdoms under domain Eukarya. So if we write the names, we have Kingdom Protista. Most of the microorganisms, those are the protozoans, come under Kingdom Protista. We will discuss that. Then we have Kingdom Fungi. Kingdom Fungi. So from the name itself, students, you all know all the fungus, different types of fungi, they belong to Kingdom Fungi. Then we have Kingdom Plantae. Kingdom Plantae. So again, all the plants belong to Kingdom Plantae. And finally, we have Kingdom Animalia. So all the animals belong to Kingdom Animalia. You all have to remember the 4 kingdoms under domain Eukarya. Kingdom Protista, Kingdom Fungi, Kingdom Plantae and Kingdom Animalia. So with that introduction, students, we will move on to the first kingdom, that is Kingdom Protista. So kingdom protista. You're familiar with the organisms that belong to kingdom protista. You all can remember, students, under microorganisms, we have discussed protozoans and algae. Both these organisms come under kingdom protista. So first, I will write the types of organisms that belong to kingdom protista. Protozoans, and algae. So you all can remember, students, under protozoans, before we go to the features of protista kingdom, we will look at the examples that you are familiar with. Now you can remember we have discussed amoeba. These are scientific names, students. So if we write it, we need to underline it. We will be discussing the standards under nomenclature, but you all are familiar with that as well. If it is printed, it will be in italics or italicized. But since I am writing it, I am going to underline them. Amoeba, then you have studied Euglena, Paramecium. These are all protozoans. These are unicellular protozoans. You can remember they have different shaped cells. Now amoeba, it has the pseudopodia for locomotion. Euglena has flagella for locomotion. Then Paramecium has cilia for locomotion. We have discussed all those properties before. And all these organisms, they are heterotrophic except Euglena. Euglena has chlorophyll, so it can produce its own food. But amoeba, Paramecium, even Plasmodium, that is also a protozoan. All of them are heterotrophic. Then we have the second group, algae. Under algae, you all can remember again, Chlamydomonas. Chlamydomonas, that is also a scientific name, that is a unicellular organism. So all these, amoeba, Euglena, Paramecium, Chlamydomonas, all these are unicellular organisms. The first three are protozoans, Chlamydomonas is algae. Then there are other algae, you all can remember, ulva. Then there is cladophora. Cladophora. All these algae you have learnt in your previous grade also. These two, this is like a filament-like structure, whereas ulva, it has a thallus-like structure. Even you can remember Spirogyra. So those are multicellular, whereas the first few, they are unicellular. But you all know Chlamydomonas, Euglena, ulva, cladophora, all of them have chlorophyll. So they produce their own food. So there are organisms that can produce their own food as well. These are some examples that you already know. So now we will look at the properties. Cellular organization, eukaryotic cellular organization, obvious because we are studying it under Domain Eukarya. What is a eukaryotic cellular organization? They have a defined nucleus. So here we can say defined nucleus. Defined nucleus. Or we can say organized nucleus is present in the cell. Then if we take the structure of organisms, unicellular or multicellular organisms. So as I told you, amoeba, Paramecium, Euglena, Chlamydomonas, those are all unicellular organism, only one cell. Whereas when we say multicellular, ulva is multicellular, that is an algae. It has a thallus structure. Most are microscopic. So obviously the protozoans, they are microscopic. They are not visible to the naked eye. Even Chlamydomonas is not visible to the naked eye, apart from the green color. They are all microorganisms. But some red algae possess large bodies. So under algae, depending on the color, there are different types. Now ulva, you all know, is a green color structure. So it's called the green algae. Like that, there are red colored algae. So they are known as red algae. Red algae possess large bodies. So they are visible to the naked eye. Then here, if we look at the specialized tissues, they don't have specialized tissues. When we say specialized tissues, they're having leaves, stem, roots, those are specialized tissues. Or vascular tissues, if you take the animals or humans, we have our skin, muscles, nerves, those are all specialized tissues. But these organisms, the protista group, that includes protozoans and algae, they don't have any tissue specialization. So all the cells are similar. There are no special types of tissues. So that is the next property we have discussed up to here. If you take the shape of organism, now ulva, you can remember students something like this. It is a stellar structure like a stellar leaf, a leaf-like structure. So you can have a leaf-like structure there, or even a horseshoe-shaped structure. Horseshoe-shaped, something like that. A horseshoe-shaped structure. And there can be different other shapes. If you can remember paramecium, that has a slipper-shaped structure. Something like that. A slipper-shaped structure. Then Chlamydomonas is something like an oval-shaped structure. So like that, they can have different shapes. So we will be again discussing these shapes and all that when we compare the different types of organisms. For now, you all need to know these characteristics or features. Then environment, mostly they live in environments associated with water. So here, associated with water. All the algae are found in water, aquatic environment. But you can remember Plasmodium is a pathogenic protozoan that lives inside the human body. So they can live in other environments, but mostly they are in environments related to water. Marine environment, fresh water, all those are the common environments. Then if we take the reproduction, they reproduce asexually. Asexual reproduction, sexual reproduction. In sexual reproduction, there are gametes. But in asexual reproduction, there are no gametes. So here they reproduce asexually. Binary fission. When we say binary fission, if there is a cell like this, if this is divided into two and two new organisms are formed, then we call it binary fission. Binary means two, fission is dividing into two. So binary fission. Then fragmentation. Now part of this ulva structure, these parts can be broken off or they can come off, separate off as fragments, and they can grow into a new organism. That is known as fragmentation. So by spore production also they can reproduce. Spores are small structures that can develop into a new plant. But this is or an organism. This is asexual reproduction. So those are different methods by which the kingdom protista organisms can reproduce. Then if we look at the nutrition. Now there are two types of nutrition. One is photosynthetic, or we can say autotrophic. Autotrophic. So if they do photosynthesis, they will have chlorophyll. You all should remember students, when we say photosynthesis, the green plants who possess chlorophyll and the algae who have chlorophyll, they both can do photosynthesis. So algae are photosynthetic, or we can say autotrophic. Then we have the unicellular protozoans. They are all heterotrophic. So they obtain their nutrition from other organisms. And you know, most of these protozoans, they not only obtain the nutrition but also cause diseases. They are pathogenic as well. So unicellular protozoans are heterotrophic and pathogenic. But the exception being Euglena. Euglena, you can remember, it has chlorophyll, so that produces its own food. You can remember all these information, students. Then types of organisms, we have the protozoans and algae. So I am sure you all can remember all these features. This is kingdom protista. So the first kingdom under domain Eukarya. Organisms with eukaryotic cellular organization can be unicellular or multicellular, can be microscopic or large bodies. Then they don't have special tissue differentiation. They can have different shapes for their body structure, and also they are usually present in aquatic environment or water environment, and then they have different types of asexual reproduction methods, and nutrition, they can be photosynthetic or heterotrophic, and the protozoans and algae belong to this group. With that introduction, students, we will move on to the next slide. So here you can see these two structures. Here this is an algae, ulva. So you can remember this is a thallus structure or thalloid body shape. So this is a thallus structure. You can remember that. Thallus or thalloid body. Thalloid body. So this is multicellular. You can remember that vessel. And it is green in color, so has chlorophyll. It can produce its own food, autotrophic or photosynthetic. On the other hand, we have a protozoan there. What is that? It is paramecium. Paramecium causes diseases. It is not photosynthetic, it is heterotrophic. So there you can see the shape of the cell. Unicellular, microscopic and heterotrophic. Paramecium. And you can remember, students, they have those cilia around their body for locomotion. So those are some of the examples. So as I told you, under algae, there are many more algae. Chlamydomonas, Pyrogyra, Cladophora, ulva, all these are different types of algae. Then under protozoa also amoeba, paramecium, euglena and even plasmodium. All of them come under protozoa. So you all have to remember their names. Remember the example, students. Now these are something that you might have seen, but you may not have identified them as algae. So what I will do now is I will show you all a video so that you can get a better idea of the different types of algae that are present and also different protozoans. Okay, students. Now you have a good understanding, or you can imagine how these algae and protozoans are. So you have a better idea about them. So those are the organisms belonging to kingdom protista. With that understanding, students, I will move on to the next slide. So here we have some useful and harmful effects of protist to human. So under that, if we look at the first effect, algae, they are primary producers in the food chains of aquatic environment. So you know all these terms. Algae is one type of protista. So they are the primary producers. Why? Because they are photosynthetic or autotrophic. You all know what primary producers are. In a food chain, always we start the first link with a primary producer. That's the first link. So from the primary producer only, the consumers get their nutrition. So algae become the primary producer in aquatic food chains or in aquatic environment. You know what food chain is. The relationship between the organisms due to its nutrition. You can start with algae, then the algae can be consumed by small fish can be consumed by large fishes, and the large fishes can even be consumed by a kingfisher or man or any other organism. So there you can have a food chain where the primary producer is algae. Then we have the second use. Algae involved in forming mutualistic associations with fungi, and that is called lichens. Now, when we say lichens, it's both algae and fungus. So, they exist together. That is what we say as mutualistic relationship. They not only exist together, they help each other. It's also known as a symbiotic relationship. They exist together and help each other. So, in that, you can see fungus, you all know students, they are all heterotrophic. They cannot produce their own food, but they can decompose and they can get the nutrition. So, fungus can help in that manner, whereas algae, they can produce their food. So, algae will produce or synthesize the food and provide it to the fungi, whereas fungi live with algae in a mutualistic association known as lichens. And these lichens are also very useful to us. Then if we look at the next one, to extract agar. Now when we say agar, and you can see which is used to prepare culture media to grow bacteria. Not only bacteria, even in tissue culture. So, tissue culture we will be discussing in the next lesson, where we discuss different types of reproduction methods. From a simple tissue, you can prepare or produce a lot of plants. So, for that we have the culture medium that is used, and for that we need agar. So, agar is a substance that is similar to gelatin. So, you can understand they are used to make the medium solid or to solidify the medium. So, when we make culture media, we need like a semi-solid or solid medium on which these organisms can grow. So, to make this culture media, we need to have different substances. So, nutrients have to be there. In addition to that, the media has to be solidified. For that only we use agar. And agar is extracted from algae. That is the third use. Then if we look at the next one, to extract alginic acid used to make ice cream. Now this alginic acid is obtained from a type of algae. So this is again obtained from a type of algae. Alginic acid, that again is like the solidifying substance to make ice cream to be that particular state. So, to solidify it, we use alginic acid. Then there are some protozoans that cause diseases. So, we call them as pathogenic protozoans. Pathogenic protozoans. Can you all remember these examples? Pathogenic protozoans. Amoeba causes amoebiasis. Then Plasmodium causes malaria. Then there is sleeping sickness that is also caused by a protozoan. If I write the examples here, amoebiasis, biasis. So from the name you all know students, it's caused by amoeba. Then there is malaria. Malaria is caused by Plasmodium. And also sleeping sickness. That is also caused by a protozoan. So these are different diseases caused by pathogenic protozoans. Amoebiasis, then malaria, sleeping sickness. So mostly you can see students, algae, they are very useful to us. Whereas protozoans, they are harmful to us. But all of them exist in the environment and they are required for the balance of nature. So that is Kingdom Protista. With that information students, I will move on to the next slide. So this is again for your extra knowledge. Below are some algae belong to Kingdom Protista and they are classified according to the color of them. So by just looking at the picture, you can identify them, students. Now you can see this is green in color. You can see the thallus-like structure, multicellular. So that is a green algae. So from the picture itself, you know it is a green algae. Then how about this one? What is the color? Red. So this is a red algae. We said the red algae, we said in the previous slide or the first slide actually, the red algae are usually larger in size. So they have a large body. So here you can see those are the red algae, an example of a red algae. Then on this side, what is this color? Brown color. So here these are brown algae. Then again this one, those are the free floating algae. So they also have a light brown color. These are the three main groups that you can easily identify: green algae, red algae and brown algae. So those are again examples. And you can see this is like a single cell, whereas these are all multicellular structures. So those are examples of algae with different colors. So I am sure you all would have understood this clearly. But to give you all a better understanding, we will look at a video where you can have a look at more types of algae. Okay, students. So now after watching the video, you'll have an understanding, better idea about the different types of algae. And you would have even recognized some of these, you would have seen them before. So you know what these are. With that understanding, students, I will move on to the next slide. Kingdom Fungi. So as we said, there are 4 kingdoms under Domain Eukarya. First one is Kingdom Protista. Under that we discussed algae and protozoans. Now we come to the second kingdom, Kingdom Fungi. So from the name itself, you all know the fungus belongs to this kingdom. So if we take the cellular organization, what type of cellular organization will be there? It will be eukaryotic cellular organization. That means the cells will have a defined nucleus. So then, if we look at the cellular organization, you know they are all eukaryotic cells. Then if you look at the cell structure, you're familiar with some of these examples, fungi. You're familiar with yeast, which is called the Saccharomyces. The scientific name is Saccharomyces, common name is yeast. Then mucor. Then you're familiar with mushrooms. All these are different types of fungus. And in their cells, the cell wall is made up of a substance called chitin. So we call it chitinous cell wall. Chitin, it is a polysaccharide. You're familiar with polysaccharides. We have discussed carbohydrates, under that polysaccharide. So chitin is a polysaccharide. And the cell wall of this fungus is made up of chitin. So that's why we say chitinous cell wall. Then cellular organization, eukaryotic cell. Then we look at the structure. Fungus again you know, unicellular and multicellular. What is the example for unicellular one? Yeast. Yeast is unicellular. Here yeast is unicellular, yeast or Saccharomyces. Whereas if you take mucor, they have the filamentous structure. If you take mushrooms, they are multicellular. So mushroom is multicellular. Most are microscopic. Most of them, they are microscopic, so you won't be able to see with the naked eye, but you can remember even mucor. It's visible to the naked eye. You have observed in your previous grade the mucor growing on bread. The filaments as well as the black color spores, you have observed that. So most are microscopic. And their reproductive structures can be seen with the naked eye. So here again, mushrooms. The reproductive structures are seen with the naked eye. That also you can remember. You would have seen different types of mushroom. And you all know we take mushroom as a protein supplement in food. So that's used for cooking. So that is visible with our naked eye. And there is no tissue differentiation. So as I told you, you can't differentiate them as like stem, root, leaves, any type of tissue differentiation is not there. But you can get the mycelium or the filamentous structure. You can remember thread-like structures, mycelium. Then if we consider the shape, they can be as single spherical cells or fungi mycelium. So again, single spherical cells, yeast is a single spherical cell. Whereas fungi mycelium, I told you all, mucor has mycelium, the filament-like structure. So that's what you all have to remember, students. Single spherical cell, yeast. Whereas fungi mycelium is mucor. So here it will be mucor. And single spherical cell, it will be yeast. Yeast for this one. Then if we look at the environment where they grow, environment and substrates. They live on organic matter and living body. Organic matter, why? Normally they act as decomposers. They are known as saprophytic organisms. You all can remember that. They decompose complex organic matter, that is dead animals and plants, to simple organic substances. So that is why they live on organic matter. They live in living bodies as well. You all know the fungus can cause diseases to skin. So they live on the living body, on the surface of the living organism. Then they can live in aquatic environments, but less in aquatic environments. Very few are found in water environments or aquatic environments. I am sure you all can understand all these features. Then if we look at the other properties, there are about 1.5 to 5 million species belong to this kingdom. There are about 1.5 to 5 million species that belong to this kingdom. And they contribute greatly to decompose organic matter in the environment. So you can remember that I just discussed that with you. They also form symbiotic associations with other organisms. What is the symbiotic association? We discussed with the protists. With algae, they form the lichens. So here, lichens is the symbiotic relationship. Lichens. Algae and fungus together, it is a mutualistic relationship or known as symbiotic association. So students, some of these features, as I told you before also, will be discussed again in the table that is found in your textbook. So these are the features of kingdom fungi. Then if we consider the nutrition, they are all heterotrophs. This is very important. All of them are heterotrophic and most are saprophytic organisms. So you know the meaning. Heterotrophic, they don't produce their own food. They depend on other organisms for their nutritional needs. So that is what we mean by heterotrophic. Under that, saprophytic, they decompose dead organic matter. So they live on dead bodies of the dead animals and plants and they decompose it and they satisfy their nutritional needs. So saprophytic organism. If we look at the examples, all the types of fungus come under this group. So these are the fungus. When we say fungus, I'll write some of the examples. yeast or what we call as Saccharomyces. So Saccharomyces, again a scientific name, so we need to underline that. Then there is the mushroom. An example or a name that you need to remember, Agaricus. Again a scientific name. And also we can say bread fungus. Normally we call it, that's like a common name, bread fungus. What is that? It is mucor. In mostly bread, fruits, all that, the mucor grows, the bread fungus, just a common fungus. So we have yeast, that is Saccharomyces, mushroom, Agaricus, and the bread fungus, that's mucor. These are all examples of kingdom fungi. That is, they are all different types of fungi or fungus. You all know that also. Fungus is singular, fungi is plural. So those are the features that you need to remember, students. They have a chitinous cell wall. That's very important. Then also we will be discussing about chitin in exoskeletons of arthropods. Then also I will remind this to you all. So remember, two different groups. One is the kingdom fungi, then we will have the arthropods. Both are related to chitin. Chitin is a polysaccharide. So here chitin is present in the cell wall. There you will see it's present in exoskeleton or exoskeleton made up of chitin. Then cellular organization, eukaryotic. They can be unicellular or multicellular. They can be microscopic or they can be seen with the naked eye. No tissue differentiation. Then they can be a single spherical shaped cells, or they can have the fungi mycelium. And also they mostly live on organic matter and living body and less in aquatic environments. There are about 1.5 to 5 million species, so many different types of fungi. And most of them are very important because they decompose organic matter and they involve in symbiotic relationships. And also all of them are heterotrophic. That's very important. All are heterotrophic and most of them are saprophytic organisms. These are the features that you need to remember, students. So with that, I will move on to the next slide. Again here you can see, students. This is the fungi mycelium. So you can see here, students, like the filament-like structure, like thread-like structure. You have observed this before also. Mucor growing on bread. So that is the mycelium, filament-like structure. Then if we look at this picture, here you can see these are the reproductive structures of fungi. Most of them are mushrooms. Here you can see this is like an umbrella-like structure. Normally when you would have been young, when you were small kids, you might have even played with these structures. You would have enjoyed seeing them. So there are different colors, slightly different shapes. So you can observe all those here. Now these are the reproductive structures and also these are the mushrooms that can be used as food, as protein supplement. But at the same time, there are some mushrooms that are poisonous. So you have to be careful. Some can be consumed as food, some are poisonous. So that has to be selected and taken. People who normally use mushroom, who pluck mushroom, they know what is suitable for consumption and what are poisonous. So here you can see they are very colorful and they have slightly different shapes. So those are the reproductive structures of fungi. So as I told you and as I have been showing you all videos, now also we will watch a video where you can understand how these reproductive structures, the mushrooms, grow in moist environment. Okay, students. So now you all have an understanding or idea about how these mushrooms grow. So here you can remember, students, these fungi can be found mostly in moist environment. So you were able to observe that as well. So different types of moist environment, say on a rock or soil, moist environment, but not inside water. So that is something you all would have observed before also. So with that understanding, students, we will move on to the next slide. So here we have some useful and harmful effects. Below are some useful and harmful effects of fungi to human. So first we will look at the useful effects of fungus as a supplementary for protein. So earlier also I mentioned this to you all, students. Mushrooms are supplementary for proteins. So here we have mushrooms. Example is agaricus. Agaricus is mushrooms. So you can see if it is printed, the name is italicized. Otherwise, you will have to underline it. So if you are writing the name, you have to underline. So when you say mushroom and if you write agaricus, you have to underline the name. The same way, so that is the supplementary for protein, agaricus or mushrooms. Then bread and alcohol fermentation. You are familiar with this. You have observed this activity also. You take a container with sugar solution. Into that you put yeast and you allow it to be there for a few hours and you observe. You can see the fermentation process. Or you have even done it with a bottle. Inside the bottle you take sugar solution. Into that you add yeast and you observe the fermentation process. You can even fix a balloon to that. You can observe the inflation of the balloon. All those activities can be used to explain fermentation. So for the production of bread and also production of alcohol, we use fermentation, and for that we used yeast. Yeast is Saccharomyces. Saccharomyces cerevisiae. You can remember that name, yeast, Saccharomyces. Then to produce antibiotics, you have studied that also before. Penicillin, amoxicillin. Like that, there are different types of antibiotics. Now, what are antibiotics? Those are chemical substances produced by one type of microorganisms in order to retard or destroy another microorganism. So normally fungi produce antibiotics or the chemical substances against bacteria. So an example is penicillin. From the fungus Penicillium. Penicillium fungus is used to produce penicillin. Penicillin is the antibiotic. There are many different antibiotics. You all remember those examples as well. So penicillin is obtained from Penicillium fungus. Then they decompose dead bodies and structures. Decomposition of dead bodies and structures. So what do we call them? We call them as saprophytes. Saprophytes. Decomposition of dead bodies and structures. Those are the useful effects. So what is the use of decomposition? If the dead bodies remain in the environment, that can pollute the environment. That is prevented. And also when they are decomposed, all the nutrients, the simple organic compounds, the minerals, they go back to the soil. So the soil becomes fertile. So they help in the cycling of minerals. Different elements and minerals are cycled in the environment. We will be discussing that in another lesson in the next year. But here you can remember they are useful for the environment. So those are the useful effects. Then we have the harmful effects. Harmful effects. So under that, if we look at the first one, they cause diseases to plants and animals. So, although we said harmful and useful effects to humans, now when they cause diseases to plants and animals, that can indirectly affect us because we have plants that are grown as crops, then animals in the farms. So, when they are affected by these diseases, that affects us as well. So, they cause diseases to plants and animals. So, Candida actually causes pityriasis or aluhum, what we call as aluhum. So, Candida is a fungus that causes aluhum or what we call as pityriasis. So, from the name, you all can understand, students, it is a disease caused to the skin and you get grey color patches on the skin. So, that is caused by the fungus Candida. Then you are familiar with potato late blight. Potato late blight. What happens there? Due to the fungal infection, initially there are brown colored dots or patches forming on the potato plant, and later it becomes black in color and finally it affects the whole plant. So that is potato late blight. So Candida is the fungus that causes aluhum or pityriasis, a disease related to the skin. But again, that also has other effects as well. There are other symptoms. And also potato late blight, again a disease caused to the plants. That is one harmful effect. Then we have the second harmful effect, spoilage of food. That also you have observed. Now spoilage of food on bread, fruits, even grains, all these can be affected by fungi. Vegetables, they can also be spoiled by fungus. Especially food that have moisture in them. Mostly moist food is spoiled. Moist food. You all know for the growth of microorganism, moisture is very important. So when the food is more moist, there's more tendency for the fungus to grow on it. So that you have observed before. So these are the useful and harmful effects, students. You have to remember them. They can be used as supplementary for proteins, that is mushroom, agaricus, then bread and alcohol fermentation, yeast or Saccharomyces, the same organism. Then to produce antibiotics, an example is penicillin from the fungus Penicillium. Then they are saprophytes, so they decompose dead bodies and structures. They are useful for the environment. Then the harmful effects causing diseases, so then they act as pathogens. Pathogens or pathogenic fungus. So an example is aluhum or pityriasis caused by Candida fungus, and spoilage of food, that is when the food is more moist. So these are some harmful and useful effects of fungus. With that understanding, students, I will move on to the next slide. So here we will do this activity in order to observe mucor on a slice of bread. So activity, let us observe mucor on a slice of bread. To do this activity, these are the required materials. We need a slice of bread. We need water. As I said, for the growth of fungus, there needs to be water or moisture. Then we need a light microscope. A light microscope. Here we need to have glass slides. Glass slides. You all know, students, to observe something under the light microscope, you need to mount the specimen onto a glass slide. And we need to cover it with a cover slip. So these are the materials. We need a slice of bread, we need water, light microscope, and we need glass slide. So let us look at the method of this activity and then we will do the activity. So method, add a few drops of water onto a slice of bread and leave it for 2 days. Why do you need to leave it for 2 days? So that the fungus will grow on it, mucor will grow on it. Then you will see a mycelium of fungi. You will see the white color, thread-like structure, and also you will see black color spots, black color areas. So that is the growth of fungus mucor. And then what we do is we obtain a small amount of the mycelium using a glass rod, put it onto a glass slide, cover it with a cover slip and observe through the microscope. So we can observe it under the light microscope. You will be able to observe the mycelium structure more clearly. So that's a very simple activity, students. You are familiar with this process. Let's do this in the lab. Okay, students. So now we are going to observe a special type of or a certain type of microorganism, that is mucor, a type of fungus that grows on bread. So I have already prepared a slice of bread. I sprayed water and left it aside for 3 days. Now here you can see the growth of the fungus, mucor. So from this we have to take a clean glass slide I have here, and from here I have to take a small sample of the fungus, the piece of bread with the fungal growth, and place it on the glass slide, and then I will have to put a drop of water on it so that you can easily cover it with a cover slip. And here again I have a clean cover slip also. So we place that, and now we use it to observe under the light microscope. So I'll remove whatever slide we have. We have to put it to low power, and then here if I make sure that the sample is in place with the light source. So now if we observe it through low power, can you all see the growth of fungus there? You can see that particular type of microorganism grown on bread. Can you all identify the shape of it? Yes. So if we observe this through the microscope, you can confirm that microorganisms, that is a fungus which is called mucor, has grown on bread. The same thing can be observed under medium power also. So if I adjust it there, now can you all see it more clearly? Can you identify the shape of the microorganism? Yes. So there you saw what mucor is, and you know it grows on bread. Okay, students. So you all were able to observe the fungus mucor that grew on a piece of bread. So this is how we saw the piece of bread after spraying some water onto it and leaving it for 2 days. You will see the growth of the mycelium there. So this is the mycelium grown on bread, and here you can see the black color spots also, white and black. So mycelium is basically the filamentous structure, that is mucor that grows on bread. So then we took a small sample from that. We observed it under the light microscope. You all know how to mount the slide. Put a sample of the fungus, put a drop of water, cover it with a cover slip without trapping air bubbles, and then we observed it under the light microscope. And you all were able to observe the fungus mucor under the light microscope. Light microscope. So that is the mycelium structure. So fungi can exist as unicellular organism or as mycelium. So with that understanding, students, I will move on to the next slide. So here, as I told you all before, we are going to compare the 3 different groups. So the first group is bacteria. So that is domain bacteria. Domain bacteria. Then we have protista. Protista is a kingdom under domain Eukarya. So this is kingdom protista. Protista. And this one is fungi. So kingdom fungi. Fungi. Both of these come under domain Eukarya. So under the 3-domain classification, first one is domain archaea. Under that it's the methanogens and halophiles. Then we have domain bacteria. Under that the bacteria and cyanobacteria. Then we have kingdom protista. Under that we have algae and protozoa. Then under kingdom fungi we have only the fungus. So these two come under domain Eukarya. So here the table is to compare the properties of these different organisms. So as I told you before also students, I said when we were discussing the properties that I will discuss some of the properties again with this table. So there are also I had included some additional properties. We will be discussing them also here. So under this, the first feature is the structure of the organism. When you look at bacteria, they are all microscopic. That is one feature. Then they are unicellular and also prokaryotic organism. So microscopic, you cannot see with the naked eye. Unicellular, one cell. And prokaryotic, the cell does not have an organized or defined nucleus. So those are the properties for bacteria under structure. Then if we take the protista, again they are most microscopic. They are also microscopic, but you can remember red algae, they possess large bodies. So they can be microscopic or they can have a large body. In one of the slides you were able to see the green algae, brown algae and the red algae. So some of them they possess large bodies. Even ulva, you can remember it has a thallus structure. And they are all eukaryotes. So they have a cell with a defined or organized nucleus. Then mostly they can be unicellular. So when we say mostly they are unicellular, you can remember students, amoeba, paramecium, euglena, plasmodium, all these are protozoans. They are all unicellular structures. Then under algae also, if you take the chlamydomonas, they are again unicellular structures. So most of them are unicellular, but some are multicellular without a tissue differentiation. So that is where we mentioned the thalloid structure. You all can remember. Thalloid structure. No tissue differentiation, but they have a multicellular large body. So that is the structure of protista. Then if we take fungi, most of them are again microscopic, but some reproductive structures can be seen with the naked eye. So here it's very important students, only the reproductive structures. Fungus, mainly the reproductive structures are seen with the naked eye. As an example, you can consider mushroom. They are eukaryotic. You all know the spelling here students, eukaryotic. I will write it here. Eukaryotic organism, eukaryotes. So they are eukaryotic organisms. So you have to remember the spelling correctly. eukaryotic, eukaryotic organisms. Here also you can see the spelling. So then if you look at the features, some are unicellular. Some of these are unicellular organisms. So here they are unicellular and some are multicellular. So when it comes to Saccharomyces or yeast, that is unicellular. Whereas mushrooms with large reproductive structures or even mucor, they are all multicellular. and they exist as multicellular mycelium. What is mycelium? The filament-like or thread-like structures. And again, there is no tissue differentiation. So you can understand, students. From this table, we are not only looking at the differences, we can understand the similarities as well. Differences and similarities among the three groups of organisms. So that is the first character of each structure. I will move on to the next character. So the next one is shape. So here again, students, you all can see bacteria, they have different shapes. So if we consider the shape, first one, a spherical shape or what we call as coccus, they are like this. Can you remember streptococcus? So their coccus means the shape is spherical. When we say streptococcus, many circle-shaped bacteria exist together like a tape-like structure. Streptococcus together. So that is spherical shape. Then there is the bacillus shape. Bacillus is like a oval shape or a rectangle type of shape, bacillus. Then we call it the rod shaped or bacillus. Then there is the spiral shape. Spiral shape, something like that. Spiral shaped organism, that is called the spirillum. That is again a bacteria. Then there is the comma shaped one, something like a comma like this. That is called the vibrio. So there is the spherical shape, coccus, rod shaped is bacillus, spiral shaped is spirillum, and comma shaped is vibrio. Those are the other names given to these four different shapes. Coccus, bacillus, spirillum, and vibrio. So are the different shapes of bacteria. So that is only for bacteria. You all have to remember that. Then if you take the cyanobacteria, they also exist as single cells. They can be branched or they can be unbranched filaments. So cyanobacteria can be single cells, branched or unbranched filaments. So that is a different type of body structure. So under bacteria itself, you can see all the bacteria, they are unicellular. Cyanobacteria can be unicellular or multicellular filaments. Can you all understand that? Then if we look at the protista. So again, under protista, there are protozoans and algae. So if you can remember, students, the leaf-like structure, protista. Ulva has a leaf-like structure. So you can remember the shape of ulva, something like this. A thallus body shape that looks like a leaf, something like a salad leaf you can imagine. That is ulva. Then there is the horseshoe shape. Horseshoe shaped. So like this you can have the shape. That is also another shape for protista. Then as I told you before, amoeba. Amoeba has a irregular shape. You can remember amoeba with the pseudopodia. It has an irregular shape. Then paramecium has a slipper shape. Paramecium, something like that, a slipper shaped structure. So like that, they have different body shapes. You should be able to identify these organisms. Then if we take the fungi, they exist as single spherical cells. So again, single spherical cells like yeast. You can remember a single spherical shape, or they will have the fungi mycelium. This is the mycelium. Fungi mycelium. So that is the shape of the body of bacteria, protista and fungi. I am sure you all can understand that. So with that, I will move on to the next feature. So then we have the next feature, nutrition. So under nutrition, bacteria and cyanobacteria. So bacteria, they are heterotrophic. So they can be decomposers as well, and they depend on other organisms for their nutritional need. So mostly heterotrophic, especially the bacteria, they are heterotrophic. So if we take the cyanobacteria, they are autotrophic. So they can produce their own food. So you can remember, students, bacteria, they are mostly heterotrophic. And cyanobacteria, they are autotrophic. But there are some bacteria that are chemosynthetic as well. Photosynthetic, chemosynthetic bacteria are also there. So mostly heterotrophic. Cyanobacteria are autotrophic. That's how you have to remember. That's why we use the word mostly. Mostly heterotrophic, mostly decomposes, but sometimes they can produce their own food. Then we have the algae under protista. You all know they are all photoautotrophic. Photoautotrophic. So they produce their own food in the presence of light using chlorophyll and carbon dioxide and water from the environment. So they are all photosynthetic algae or phototrophic algae. Photoautotrophic algae. Any term can be used there. So they basically produce their own food. Whereas if you take the unicellular animals, there you can see mostly the protozoans. So here protozoans are heterotrophic. Protozoans, they depend on other organisms for their nutritional need, and also you can remember they are mostly pathogenic. They cause diseases as well. So algae are phototrophic or photoautotrophic, or you can say autotrophic organisms. All terms can be used. Whereas protozoans, those are the unicellular animals, they are heterotrophic. Then if we look at fungi, they are all heterotrophic. That is important. All of them are heterotrophic. And most of them obtain nutrition as saprophytic organisms. So what is the meaning of saprophytic? They live on dead animals and plants, bodies of dead animals and plants, and they decompose them. So that is saprophytic organism. So all the fungi are heterotrophic and they obtain nutrition as saprophytic organisms. So you can see here, students, bacteria and protista, you can have autotrophic or heterotrophic organism. Whereas fungi, all are heterotrophic. That's how you have to compare and understand. There are similarities and differences. So with that, students, I will move on to the next slide, where we look at the feature reproduction. I have explained some of these methods before also. So first we will look at bacteria. They mostly perform asexual reproduction. So under that, they can do binary fission. That is one method. So bacteria normally binary fission. Whereas cyanobacteria, they reproduce by fragmentation and budding. So if we look at this first type, I have explained this before also. Now let's say if you have a bacillus structure, a rod-like structure. Now this divides into two, forming two new bacillus structures. So that is known as binary fission. So one cell divides into two new cells, binary fission, seen in bacteria. Whereas in cyanobacteria, it's fragmentation. You can remember cyanobacteria exist as filaments. So if we have, this is the first type, then if we take the second type, if there is a filament, now this can be broken or it can separate into fragments. The fragments need not be of the same size. They can be different sizes as well. So that will give rise to one new structure that will further grow into the filament, and the second one also will grow into the new organism. So one filament can break or separate into many fragments. That is fragmentation. Fragmentation. Even the next one is budding. Now, if you take the third one, buds are like from this particular cell, there is a small, like a protrusion. That's a bud. This particular protrusion is a bud. And that bud separates from the main organism and it grows into a new organism. An asexual part that can grow and develop into a new organism. So that is the bud. So we have binary fission, fragmentation and budding. And also under protista you can see there are spore production. You can remember students even under fungus. Fungus, they have the mycelium as well as spores. Spores are again simple spherical structures. So another type, we can say spores. Spores are small spherical structures like that. That again develops into a new organism. That type of reproduction is also there. So binary fission, fragmentation and spore production. So we will look at those three separately again. So bacteria, as I told you, bacteria reproduce by binary fission, cyanobacteria by fragmentation and budding. The same way if you take protista, again that is also asexual reproduction. Under that they have binary fission, that is one. Then they have fragmentation and the third type they have spore production. So that is three different types of asexual reproduction there. Whereas if you take fungi, now fungi only reproduce asexually by spores. Only one type of reproduction. So now also you all can understand, students. Compare and understand. There are different types of sexual reproduction. So I am sure you can understand that. So with that, students, I'll move on to the next slide. Here again we have distribution. So when we say distribution, where you can find these organisms. If you take bacteria, they can be widely distributed in air, water, soil and in the body of organisms. So inside and outside as well. So air, water, soil and in the body of organisms. You can remember normally they come under microorganisms. They are basically found everywhere. When we discuss bacteria also, I told you all they are found everywhere in the environment. And so here also it says almost everywhere. So the examples are bacteria. So here also students, bacteria. This is the table from your textbook, bacteria and cyanobacteria. Those are the examples under domain bacteria. Then we have kingdom protista under domain Eukarya. These are again eukaryotic cells. You can remember under that there are the protozoans as well as the algae. So if you look at the distribution, they can be found in the marine environment. They can be found in the freshwater environment. Usually algae are found in aquatic environment. So marine and freshwater together, you can remember the term aquatic environment. So that is algae. Then you can get them in damp soils, the protozoans, and inside the body of organisms. So that is the protozoans. You can remember that. So as examples, you can see students, algae, those are the aquatic ones. Then we have the unicellular animals. So here, this is for the next one, example. So the unicellular animals are the protozoans. So these are the protozoans. Protozoans, amoeba and paramecium. Those are all examples for protista. Then we have fungi there. Again distribution, you can see live on organic matter and living body. So we said they are mostly saprophytic. So they live on organic matter, decompose them, decomposes. And also they live in living body. Why they cause diseases? We discussed the diseases. Candida fungus causes aluhum or pityriasis. So that is on humans. Then they cause potato lead blight. So live in a potato plant. So they live in living body. but they are less in aquatic environments. They are not really found in aquatic environments. Although they need moisture for the growth, they are not found very much in aquatic environments. So, less in aquatic environment. So, these are the different features of organisms that you all need to remember, students. So, with that, I have almost come to the end of this chapter. In the next slide, I have a few organisms that belong to all the groups that we have studied so far. So, we are discussing the 3-domain classification by Carl Woese. Under that, we discuss Domain Eukarya, where there is the organisms, methanogens and halophiles. Then Domain Bacteria, under that there is bacteria and cyanobacteria. And thereafter, under Domain Eukarya, we have discussed the Kingdom Protista, under Kingdom Protista, algae as well as protozoans, and also Kingdom Fungi, which includes different types of fungus. So, if you are given the diagrams or pictures of these organisms, you should be able to identify them. We have been looking at some videos as well. So, we will go to the next slide and try to identify the organisms given there. So, identify the organisms given below. You just need to identify the group to which the organism belongs. So, look at the first picture. What is that? Can you identify the organism there? This you can see is a bacteria. So, here you know it is a bacteria. Then the second one, unicellular, this particular structure, you can see the cilia also. You know that is paramecium. So, it belongs to protista because it's a protozoan. It belongs to protista. Kingdom protista. Then we have a mushroom there. You can easily identify that. So, when we say mushroom, it's agaricus. You know the name also now, but you can identify it as a fungus. Then how about this one? You have seen this before. Euglena. Again, that is a protista. So, it belongs to group Kingdom Protista. So, this is also protista. Then what is this structure? You all can remember it is a cyanobacteria. So, it belongs to group or domain bacteria. It is a cyanobacteria. Cyanobacteria. So, belongs to domain bacteria. That also you know. Then we have this one on a piece of bread. What is that? This is mucor growing on a piece of bread. So, you know again this is fungus. You can also write it as Kingdom Fungi under Domain Eukarya. Then what is this? You can easily identify this, a unicellular organism with pseudopodia, amoeba. So, that is also a protozoan coming under protista, Kingdom Protista. What is this? Ulva. Ulva is an algae. Thallus structure, you can identify the thallus structure. So, it's an algae. Again, that also comes under Kingdom Protista. So, this is an algae coming under Kingdom Protista. So, depending on what they ask you to identify. Here, of course, some of them you know by name. So, you know ulva, you know amoeba, you know paramecium, then mucor, euglena, agaricus. For all those, you know the specific name, the name of the species. But here, you know this as bacteria, that is cyanobacteria. So, you can either identify the organism, then they might ask you to identify the type. For example, paramecium and amoeba, they are protozoans, whereas ulva is an algae. Then there we have cyanobacteria. So, that is the type of organism. They can also ask you to identify the kingdom. Kingdom fungi, kingdom protista, like that you can identify. They can also ask you to identify the domain. that also you can do. So this is just a simple practice, students. You will come across these types of questions in your exam papers as well. So with that, I have come to the end of this chapter. So in this chapter, we discuss two main kingdoms under Domain Eukarya. There are two more kingdoms, Kingdom Plantae and Kingdom Animalia. So in the next chapter, I will discuss Kingdom Plantae.